I'll just tell everyone to keep on checking the Mlcb website in case they're having problems.
Yeah, I'm just trying to reset the. I forgot to. I didn't realize you can't do that.
Hey, everyone.
I'll just tell everyone to keep on checking the Mlcb website in case they're having problems.
Yeah, I was just trying to reset the. I forgot to. I didn't realize you can't. Okay, I've got the new link. Let me just send it to you. Sir, I just sent you the link by email and also in the chat.
Okay, I will put it on the website and I'll just mention to everyone.
Thanks.
Do we want to tweet the link?
Yeah, I tweeted the wrong one. I'm going to tweet the right one.
You did?
I'm
You did,
gonna,
Aisha?
yeah,
Okay.
I'm just going to do
Let
that
me
right now.
retweet. Uh-huh.
Okay. All right, that should be done. Let me try to add the blue sky timer as well. I was able to do it, but it shut down the webinar.
David, do you want to try to share your screen and after that I'll share mine? I just want to make sure that everything is fine.
Sure.
Perfect.
Cool. So unshare and pass over to you.
Yeah.
Okay.
Okay, I think it's 9 o'clock and we should get started. Anshul, how are we doing on the number of participants?
Yeah, we're doing pretty good. We're up to 94.
Okay, so let's get started so we don't fall behind. So good morning and good afternoon, everybody, depending on where you are. On behalf of the organizing committee, we're really excited to welcome you to the third annual meeting of Machine Learning in Computational Biology, or MLCB. Just as a housekeeping note, the link to YouTube, if you want to watch it streaming, has changed and the website has been updated. So make sure you check the website anytime if you drop out of Zoom, and you can log back in, and we'll keep on continuously updating if anything happens. Okay, so before we get started with our first keynote, there was a few introductory remarks that we wanted to share with you. So this is our third annual meeting of MLCB. Since MLCB became a conference. But of course, as many of you know, MLCB has a long history, but it was conceived in 2004 as a neuro workshop. And since then, the interest at the intersection of machine learning and computational biology increasingly grew. And at some point, it became too big to be just a workshop at NIPS. And we were generally told to think of some new ideas. And this is when MLCB went through a period of soul searching in 2018, and it was reborn as an independent conference in 2019. And since then, we've seen a major increase in number of participants and the diversity of the backgrounds of participants. So similarly, we've also seen an increase in the number of submissions. There was a small dip, probably because of pandemic, but over the last few years, we've been getting around 100 really strong submissions, among which their 10 to 15 top reviewed submissions are selected for oral presentations, and you'll get to hear some of these today and tomorrow. So it's always fun to see what's changed and what remains the same. These are word clouds from abstracts and titles that were submitted in 2004 on the right and 2021 this year. And quickly squinting, you'll see that there's a lot of similarity in themes. So this is kind of cool. A lot of the problems are unsolved, and that's why we're all here still. But there's also some expected differences. I'm just highlighting through here. So there's now the emerging area of single cell genomic and lots of methods that are being developed. There's a much wider use of deep neural network, whereas it was kernel methods back in 2004. And also there seems to be a more data-centric approach to problem formulation and analysis. So we're very thankful for the very strong group of program committee members that help us review the submissions each year. And this the pool of program committee has really increased since a few years ago. We're also very thankful to the area chairs for their timely work and thorough reviews of all the submissions that allow us to provide reviews in a 2-week period between submission deadlines and notification of decisions. So let's get started with today. You can find more information on the MLCB website. Just as highlights, we're going to hear from 3 keynote speakers today and tomorrow. There's going to be 16 oral presentations, 21 spotlight talks, 3 minutes each that are happening tomorrow, followed by a poster session. And new this year, we have an industry panel tomorrow, 9 to 10 a.m. And these are all Pacific time. So industry panel is new this year, and this should be really exciting. So make sure that you join for this as well. So with that, I'm very excited to introduce our first keynote speaker today, Dr. David Kelly. David is a principal investigator at Calico Life Sciences, where he has been since 2016. So David earned his PhD in computer science with Steven Salzberg at University of Maryland College Park, and followed that with a postdoctoral fellowship at Harvard Medical School with John Rinn. After that, he was recruited to Calico. Over the last few years, David has made many important contributions at the intersection of machine learning and computational biology. As some highlights, he developed the Basset method that many of you know, and this helped pioneer a whole new approach to modeling the relationship between genomic sequence and regulatory function. The methods that he's developed and continue to develop push the boundaries of scalability and prediction accuracy of deep neural network in regulatory genomics. Personally, I'm a huge fan of David's work, and many of his papers are required reading for the courses that I teach and in our journal club. So we're very excited to welcome David. Please take it from here.
Thank you so much, Sarah. All right, let's see. Okay, does that look good? All right, well, thank you so much to the organizers for the opportunity to speak today. This really is a wonderful conference that I've got to participate in for several years now. And yeah, I just find it to be an excellent venue for discussing some of the more technical aspects of the machine learning that we all do towards biological ends. And you can write a little bit of a different project for this versus genome biology, for instance. Okay, nevertheless, I'm going to start with some biological justification for the work that we do. So, I find it to be a really amazing thing going on in the field right now. There's this incredible opportunity offered to us by human genetics through increasingly large biobanks that have genotypes measured, as well as increasingly specific and detailed and rich phenotypes. And that's given us the ability to find thousands of locations across the genome where there's a genetic association between causal variants and some sort of phenotype relationship. So all of these are an opportunity to really learn about the biology in a unique way, where you have this knowledge that there's a causal relationship. One variant does this, one variant does that. However, a lot of these associations are ambiguous in that causal variant is hiding within a set of variants that are very correlated within the population, and just the genotype-phenotype matrix don't reveal to you all the information that you really desire. In addition to that, a lot of these for complex traits are coming out as non-coding, in that there's no evidence that there's a coding mutation that would pinpoint a particular gene. So we can look at our Manhattan plot, see a lot of genes, a lot of variants, and start to feel somewhat lost. So the goals that I would lay forth for a genome-wide association study, the post-analysis of it, is for each of these associations, we want to identify causal variants for each association. We want to determine the mechanism by which the variant results in the observed phenotypes. For a non-coding set of variants, we, at this point, assume these involve gene regulation. So in that case, we want to know what gene is affected, in which cell types and situation is expression modified. And you might also want to know, if there's a variant modulating something in the DNA or RNA, what are the regulatory factors that are involved? Is there a certain transcription factor binding site, say, that's affected? All of this would then add up to, maybe not certainty about what's going on, but at least a really good set of hypotheses to go forward. So the resource that others and I have become very interested in building then is basically a big map, all of the regulatory elements in the genome, as well as links to their target genes, and of course, having that cell type specific resolution. So I wanted to say, I gave you one path of motivation to this resource through genetic associations, but I could give you a whole other. I think this is a really basic genomics resource that would have a lot of benefits just towards understanding better how gene regulation works. If you were interested in differences between species and how regulatory elements are involved in evolution, then I think you'd also want to build a resource just like this. So my path here, of which there are several, and other people are taking some different paths, and hopefully there will be great synergy between them. My path is to attempt to build models that can predict gene expression from DNA. I'll try to sell you on why that's a meaningful path. So the machine learning problem, as I've framed it over several years here, is that we have thousands and thousands of functional genomics data sets that describe regulatory activity across the genome. We have a variety of histone modifications. We have accessibility, transcription factor binding sites. And I'm going to talk a little bit in this talk about CAGE, which is a method to measure gene expression from the 5' end of the transcript. So it really highlights these active transcription start sites. Okay, so the machine learning problem here at Frame is we have all these data, you have the DNA sequence underlying these data. So can we look at some of the chromosomes, look at a variety of these DNA sequences to train a model that when it sees a new sequence is able to predict all of these functional attributes. Now, at this point, you would be able to make a compelling argument that you understand and can read the DNA of the genome if you can sort of impute all of these different regulatory activity annotations. So the main tool that I've found is quite useful for this task is the convolutional neural network. When it's operating on a DNA sequence, the first layer of this network has these convolution filters that are able to capture transcription factor motifs and other nucleotide composition elements of the DNA sequence. This is all something we're relatively comfortable with. These structures have been around in bioinformatics for a long time. After that, it gets a little wild. We're hitting these with nonlinear activation functions. We're maybe using normalization layers. We're pooling to collect information. Ultimately, we have vectors that represent segments of sequence, which we hope contain all of the information that you would need to make good predictions about that sequence. So if we want to think about large regions of sequence, and we want to be able to reason about distant interactions like an enhancer that influences expression from a promoter that might be tens of thousands of base pairs away, then we need to set up the model so that a very long sequence is coming in so that we're doing these convolutions to pool to a resolution that is meaningful for us to be able to make predictions. For all the work I'm going to show you, I've chosen 128 base pairs as that resolution, but we'll discuss resolution and pros and cons a little more at the end. And then you need some sort of module to share this information across a sequence. So at this point, each of these are vectors here, each position across the sequence that represent some region in the genome. And the top here is just a cartoon. The model doesn't know these annotations, but we hope that these vectors have learned, I look like a promoter, I look like an enhancer, I look like a CTCF site. The task of this third block here is to tell the promoter what is surrounding it and how should it change how it feels about its activity level in lots of different cell types based on seeing these other regulatory elements around. So if you're somewhat familiar with deep learning, you might come in immediately with some hypotheses about different types of blocks that would be effective here. And I'm not going to necessarily say any others are wrong at this point, but in the first attempt I made at this problem, what I found what worked best for me was called dilated convolutions. Dilated convolutions are very analogous to the convolution. In fact, in most of the deep learning software, it's the exact same method. You just change one of the arguments, and you change the argument to specify that each position in the convolution is going to have a certain number of gaps. So it's going to be a certain distance away from the center position. And that allows you to, when you stack these with increasing distance, and typically you increase this by a multiplicative factor, that allows you to capture this ability to share information across the sequence. So the center here, the promoter, even though its dilated convolutions are hitting certain positions that are far away rather than all positions, those positions have already looked at their region. And so you do have this ability to have gradient information flow in a fairly dense way. Ultimately, then, these dilated convolutions do their job, the vectors are updated, and we're going to try to make predictions in these 128 base pair bins for all of the data sets that I laid out. And in this original paper, it was everything I could get from ENCODE, Epigenomics Roadmap, and the Phantom Consortium. So there's thousands of these data sets. Okay. Okay, so here's that same sequence. Originally, I just showed you the data. Now I'm going to show you the data and predictions made by the model. this is a held out sequence, so it's not something we trained on. And you could just broadly see that qualitatively, this is producing meaningful results. We're able to pinpoint the promoter and some of the other accessible regulatory elements that are around. However, there is a shortcoming of this procedure, this type of deep learning block, which is that ultimately, if I pop back over here, the dilated convolutions are really fast, really efficient, and capable of sharing information in my setup for about 20 kb range. But that 20 kb range is a lot less than the distance that a lot of known promoter enhancer interactions occur at. And so after we finished this project, it was still top of my goal list to be able to find something that was able to work with longer sequences and capture interactions at greater distances. Okay, so meanwhile, over at Google Brain, our parent company, and then suddenly everywhere throughout the ML community, it seems, people started playing around with this different deep learning block that is either called self-attention or is often called the transformer layer. So what is that exactly? I should say it started in natural language processing, which is a really nice analog for a lot of the problems we work on, because you have 1D sequences. They have 1D, we have 1D. They have these words in a certain language. We have DNA nucleotides, but they immediately go to an embedding space of those words. And through our convolutions, we immediately go to an embedding space. So it ends up being somewhat similar. So it's a great place to steal ideas. This is, a secret trick of doing machine learning in compile well is if you just pay attention to what the fields that are 100 times bigger than we are do, you can usually come up with some good ideas. So the attention works as follows. You're going to take your input, which is specified as these A components here. So you have vectors across the sequence. You're going to map it to these 3 other vector spaces. They're called keys, queries, and values. The keys represent who am I? So what information do I as a position in this sequence contain? The queries specify what am I looking for? And the values specify what do I have to say? So, okay, how are these going to come together? So the queries and keys have a nice pairing. They specify what each position is looking for and what it represents. So we can take these and do dot products between all of the pairs of them to form what's called an attention matrix. So the value in this attention matrix for each IJ combination is saying, how much does position J look like something that position I really wants to pay attention to? Then you can normalize the rows of this attention matrix and treat it as coefficients to do a linear combination of these value vectors. So once I've decided which positions I want to pay attention to, then I use that to weight these value vectors of what they have to say. So the intuition here for natural language processing is that there are just words and sentences that are more closely, they're just more important to each other. They have more ability to influence semantic meaning, and therefore you want to give each of the words an opportunity to look around and exchange information. So, you're going to have high attention between, say, verbs and direct objects. You're going to have high attention between adjectives and the direct objects, but some of the other words maybe aren't as meaningful towards changing the vector at that position. Excuse me. So that's working in natural language processing space. Let's think about this in the genomic space. So I think it's easiest to think about these promoter regions where the cage signal is going to be highest, where I'd really like to be able to predict cell type specific expression with as great accuracy and specificity as possible. So you come in with a vector that says this looks like a promoter. You come into this self-attention layer, and it's going to have an opportunity to look at every other position across the sequence. So first of all, this receptive field challenge is now resolved. We're not limited to 20 kb. We're not limited to, we're limited only by the size of the sequence that we can bring into the model that fits in the RAM of your processor. So that promoter is going to say, you know, my query is I'm looking for enhancers, for example. And then all of the other regulatory elements around are going to have key vectors that specify whether they are sufficiently interesting to influence expression at the promoter. So you might have something like this, where there's a element that's pretty close, let's call this 5 kb upstream, could influence the promoter, but the convolutional filters, they don't see anything interesting. And so this content-based comparison to compute the self-attention is going to come up with a low value. Alternatively, you have this enhancer element that's way far away, all the way downstream, 50, 60 kb away. Even though it's far, we have the opportunity to look at the content of that enhancer element. And then this dot product is going to come up with a high value. And we will have an opportunity to then exchange information. So if this is enhancer that's specific to the liver, now the promoter can update its vector and say, I think I'm going to be expressed at a little bit higher value than I thought before, because I've now seen this enhancer. Okay. So we formalized this model in collaboration with Ziga Abchek at DeepMind. Vikram Agarwal, also at Calico, was a major contributor to this project. So at the top here, you can see this receptive field change. The previous model, which I called Basenji2, is limited to 20 kb. The new model, which we call the Informer, because it became very popular in the natural language processing field to use the suffix former for everything, because they really like the word transformer. And we're really interested in enhancers. So we created the Informers combination of the two. It's okay, you know. So then the convolutional layers are going to be very similar, and then we're going to have multiple layers of these transformer, also called self-attention blocks that I just described. So there's many, many opportunities to do this. Look around, I'm looking for a certain type of thing. So it's not the case that the keys queries have to immediately in one go figure it all out. They have many opportunities to say, okay, this block, I'm looking for this block, I'm looking for that. That's why you benefit from having many layers. And then ultimately, all of these parameters are going to be shared, but then I'm going to have two separate final linear transformations on the bottom of this model. One is going to make predictions for all of my human data, and one is going to make predictions for mouse data. So just a quick aside there, in the time in between publishing the original model and publishing the Informer self-attention model, I started playing around with the idea that you could use data from multiple genomes to benefit your accuracy on held out sequences, even if you were primarily interested in human. And it was the scheme that I came up with where, like I said, all the parameters are shared, and you're really just relying on the fact that across basically the whole mammalian tree, it seems, the regulatory factors and the principles by which they work, all the transcription factor motifs are, the core is really shared. There's a lot of differences, you know, there's just like tons of zinc finger proteins in humans that aren't conserved across other species. But for the main developmentally relevant transcription factors, there's enough shared that you can do this procedure where if you train on human data alone, you get one level of accuracy. If you train on human and mouse data together, just go in batch by batch. Give me some human, give me some mouse. Then there's this kind of nice generalization benefit. You see less overfitting, and you see slightly greater prediction accuracy on whichever species you're looking at from the jointly trained model. And you know, I mentioned briefly the opportunity to study regulatory evolution with these models. It's not required to use a training scheme like this to do that, but if you're interested in that, I would check out this paper because it did a bunch of analyses looking at like cross-species prediction, and thought that was fun to do. Okay, one other note on the informer model is that, so far I haven't touched at all on the distance between elements and its role in influencing the predictions. I've primarily focused on what's called content-based attention, which is mapping those vectors to vector spaces based on the vector representations and exchanging information accordingly. But we know from having studied enhancers for a long time that, if you have the same strength of an enhancer and it's at 20 kb versus 200 kb, you're going to have a very different influence on gene expression. So a really important piece of information that we have to give this model, and an inductive bias to give it to make use of, is that things that are closer together are more likely to matter. So a lot of the projects in the natural language processing field don't give this as much focus because they're typically working with shorter sequences. If it's a sentence, then does the distance really matter as much? I don't know. But for us, it was quite important to think about this, and it was quite important to use a scheme where relative position between two elements is encoded. So we basically came up with a scheme where you come up with a set of features of the distance, which are on the left, these different functions of the difference between two positions. And then within the model, these are just hit then with a neural network layer. So the distance between two elements becomes an embedding itself and is then incorporated with the content-based attention information to form the final attention matrix. So at the end of the day, it accomplishes exactly what I said, which is that you give closer things the opportunity to have a greater impact. And so on the right here, you can see our results with several different schemes. The simple but effective is just this central mask where you just have a 0 or 1 if it's within a certain distance, and you increase that distance with some sort of multiplicative factor. So this was really important to get right. When we first tried using self-attention blocks, using vanilla code that had come out of the NLP field, we were not able to exceed previous models' performance. It was only upon really thinking through and getting right this relative positional encoding that we had the success to move forward. Okay, and here's that success in another qualitative view. So here's genome browser view of a held out sequence. In green at the top for each of these tracks is the experimental data. In orange is the previous model with a fixed-ish 20 kb resolution. And in blue is the new model where we're seeing, at least for this sequence, much better results, much better ability to recapitulate the experimental data. Quantitatively across the genome, that proved also to be true. So each of the dots here is one of the thousands of training data sets for each of these different assay types. And the x-axis is the Pearson correlation in those bins for held out sequences from the previous model. And then the y-axis is that correlation for the new model. So for some of these chromatin marks that you might think are much more influenced by only local information, the prediction accuracy is, there's a nice boost, but it's not enormous, except for some of these magic TFs. And then for the cage data, you do see this nicer boost. So this is the gene expression measurements. You'd expect there to be more influence of enhancers. And so we were happy to see that that was improved with the new model. At the bottom here, we're focusing on transcription start sites and we're focusing on the cage. So we can then use gene transcription start sites as our unit to compute these correlations. And now we're going to sort of transpose the task. We want to ask for each transcription start site, we have this vector of the real data across all the different cage samples, which is about 500-600, all sorts of different tissues and cell lines and everything. And then we have our predictions. So what we want to see here is that we are able to capture specificity across cell types. And that's these correlations on the left. And you can see that, of course, this matters more for genes that have high variance across cell types relative to some genes just don't have a ton of variance. And in that case, there's not much to look at. Okay, so next, we've made this argument that this model works better because it's better able to capture long-range information. So let's begin to query that within the model to see if it's achieving meaningful results according to some other experimental data that's available. So first off, let's just take a look at a region. This is a gene, HNRMP-A1, and you can see several tracks here that are basically querying whether or not the model is using long-range information to make its prediction. So the first track I'm going to show you here is derived from the attention matrices themselves. So we came in with this hypothesis that, okay, the attention matrix, if this position is paying attention to other positions and we can kind of aggregate that across all of the layers, then maybe that achieves a meaningful quantification of how much this transcription start site is paying attention to these other regions around. And, you know, if I rank them, maybe that's a good way to find enhancers. In the second and the bottom track here, we're going to do something that is, you know, arguably simpler, which is just to take the gradient of the model prediction in a certain cell type or summarized across multiple cell types back to, in this case, back to the 128 base pair resolution right before the transformer layers. You can go further down to the nucleotides, but now you're just, we don't need that resolution here for this experiment. So right away, you can see that while the Basenji-2 model is limited to about 20 kb, as I've said several times, the new model is not. You're able to make use of information that's quite far. So what does it mean for this to have a non-zero value? It means that the gradient is non-zero. If the representation at this position before the self-attention layers were to change up or down, then the model prediction would change up or down. So, okay, there is some influence there according to the model. So the next question is that influence correct as so far as available experimental data can tell us? So our main source of this validation information are CRISPR screens that have been performed where regulatory elements are targeted rather than the genes and their coding sequences, which is, you know, a very popular thing to do for screening. Here, we're going to send something like, say, the CRISPR-I repression machinery to regulatory elements and then measure the effect that that has on the gene's expression. There's two ways that this has been done. This has been done with single gene screens where you would pick a single gene of interest and tile a large region. And typically the papers would do more than one, but they choose them very thoughtfully. In the second case, this is done using single cell RNA-seq in a far more parallelized fashion where you target regulatory elements across the whole genome, but then the readout of gene expression is just a single cell RNA-seq. And so, you know, you can imagine due to the sparsity, you have a little less statistical power here to pick up any individual interaction, but across the genome, you find lots of significant interactions. Both of these are really interesting approaches to also be building this resource I've pitched of the big map of regulatory elements connected to their genes. And we hope to have ample synergy with these approaches in the coming years. But for now, we're going to use this purely as validation data. So we're going to use our score derived from either the attention matrix or the gradients to then rank all the regulatory elements. And then some of them are validated by these experiments as, yes, influential enhancer. And some of them are decidedly negative examples from the experiment. So that allows us to use classification metrics like area under the precision recall curve to quantify how we're doing versus random or versus some other simpler experimental-based assays. We also broke this down by distance. It's harder to predict these things at greater distance. So we thought that was a meaningful way to look at the results. So the blue tracks here are our informer model, the orange are the Basenji. You can see that we've boosted the performance versus the previous model in just about all cases. And in many of these cases, we're approaching the accuracy achieved by scores that are derived from experimental data. The light green here, the ABC score, is if you have a H3K27 acetyl experiment in this cell type, then you can use that to quantify the activity of the regulatory element. And then you hit that with a measure of the distance between the two elements, of the contact frequency, which changes with inverse distance, or if you have a high C matrix, you plug that in. And so you can see that we're competitive. Sometimes we win, sometimes we lose, but overall, we were quite content to see that because ultimately, there's a lot more that you can do with this in silico model if it's able to produce meaningful promoter enhancer predictions at the level of the best we currently have. And we're quite optimistic we'll keep making this better too. Okay, I see a little activity up here in the Q&A. Is this maybe a good time to take a question, or should I be waiting till the end?
Maybe it's a good time to take a question, especially the first one, which is a little bit more of a clarification. So the question is about the number of heads, how you decide on the number of heads. I guess, is there any biological intuition behind the number of heads for genomic sequences, or is this number empirically decided?
It is, it's a hyperparameter like many other aspects of these models. The number of heads, so I kind of glossed this over. Basically, within each of these self-attention blocks, there are a number of heads, which basically means that they're distinct mappings to queries, keys, and values within each of these blocks. And so even within one block, you have an opportunity to be looking for multiple types of things. The number of heads, it ends up being somewhat of it reduces the computational expense. And so one way to look at it is that way, that it's a kind of an approximation of the full thing. If you just increased your number of layers, maybe that would be more effective than adding heads. But we do use, I think we used 8 heads. We just kind of stuck with common hyperparameters from the NLP field. It was either 4 or 8. And I haven't done extensive experiments on this, but I think it's not a hyperparameter that the model is all that sensitive to.
Thank you, David. I think for the sake of keeping on time, what we're going to do is just keep the rest of the questions for after your talk, with the exception of if there is clarifying questions, then I'll ping you for those.
Yeah, okay. I'm staring at this one and it's interesting. So I'm going to bite off one more here. Is there any way to discern between scenarios the model needs to be improved versus something about the genome I haven't measured yet? I think that's a great question and it's a little hard to answer. Diagnosing poor performance is tricky because rarely does the experimental data give you all of the information that you need. You're getting these high-level descriptions from the assay of what's going on. So if my prediction's different and I want to know why, well, experimentally, I'd love to go in and say, mutate all the nucleotides and understand their influence. And then maybe I'd see that, oh, there's this motif for this regulatory element that my model missed, but which seems to be real. And in that case, you would maybe conclude that, like, okay, well, the model's missing something here. Maybe there's something I can do with my training data, or some way to manipulate the model. I guess I would immediately break it down into, like, are we making mistakes at the motif level, at the 100 base pair resolution level? Because then in that case, it's probably more of an issue of the data providing enough signal for the convolutions to learn it. If the mistakes we're making are things like, there's this really strong TAD boundary with CTCF sites, and there's an enhancer on the other side of it, and the model thinks the enhancer should influence it, but actually there's this complicated 3-dimensional structure in the genome that the model is not able to learn. Now I'm starting to think like, okay, either the model needs to be modified in some way. And the self-attention, since we borrowed it from NLP, they don't have topologically associating domains in English sentences. So maybe there's some way you need to modify the mathematics of the self-attention layer to consider things like that in a better way. You also might then go down the road of like, well, the model isn't being given any data that really describes things like 3D architecture. So maybe there's another path where you do use that data, even though it's a little more complicated to do. And I'll say a few more words about that at the end. Okay, or maybe I won't because we're running a little short on time. Aspirationally, I thought I might, but I will briefly say it. Okay, so I pitched the justification for this work as being able to really do more with human genetics. So let's get into that. If you have a genetic variant, you can produce a prediction for its effect across these thousands of different data sets with a simple procedure, which is just to do a forward pass with one allele and a forward pass with the other and compare the results. Often I compare the results by taking the difference between the two predicted tracks and just summing up across the whole sequence. This would be a gene agnostic version. Alternatively, you can make predictions for specific places in the genome. So if you know there's a gene you're interested in, you really want scores for that gene, then you just slice out the prediction at that gene. Here's the type of thing you can do. This is a variant done at the bottom here. The C in the reference genome is sometimes a T in individual's genome. And this was measured as an EQTL by the GTEx project in whole blood. So we might take a look at then the PBMC track from the CAGE consortium. Here we see that there is high expression of the gene NLRC5, and the informer model is able to reproduce that high expression for the gene, just looking at the whole region. When we introduce the variant, so this track here is a subtraction between the prediction for the reference and the alternative allele. We see that there's a little bit of activity around the variant, but the largest effect is over here down at the transcription start site, which is about 35 kb away. So that's neat. That's something that we would have missed with the previous model. We can learn with the new model with the extended visible range. And if we further go in, initially I just scored the variance C versus T. If you want to know more about the local region and get at this like question of mechanism of why this variant really matters, you can introduce every possible mutation around that variant. We call this an in silico saturation mutagenesis because you're saturating the sequence with mutations, measuring each of their effects. And in this case, you see that the variant matters because it participates in this motif for the SP1 transcription factor. So I'd come in with a strong hypothesis that that's what's going on. You can maybe validate that by knocking down SP1 and seeing if you see a measured effect. Okay, I should say too, this was fine mapped to be the causal variant. So, one of the challenges you face is that these are really high dimensional annotations. And so working with things like the EQTL data is a little tricky. In collaboration with Hilary Finucane and Qingbo Wang at the Broad Institute, I basically passed over a huge set of predictions for genetic variants to Qingbo, and he added them to a number of features that they had already been working with for genetic variants. So things like whether they overlap a lot of the annotations that were training data, distance to transcription start site. You can throw in things like conservation metrics if you'd like. And Qingbo set up the following task, which was now that the GTEx project has matured to the state where there are many, many associations, in enough cases, we've gotten lucky, and the linkage disequilibrium structure of the locus allows for statistical fine mapping to a very high probability single causal variant. So we can take these out, and there's about a few thousand of them, and use them as positives in a second machine learning exercise, where then we choose a set of negatives, and you can either choose this to be random if that's desirable for what you want to use it for, or you can choose them to be matched for certain properties so that you make this a difficult task for your meta predictor. Either way, you would then come in and use your favorite machine learning algorithm. We use random forests here and come up with a classifier of EQTL, causal EQTL status, given all of these annotations. And transcription start site distance is the most important thing, but a lot of the computational annotations produced by the previous model, Basenji, were very valuable to getting higher performance on this task. So here, I'm going to put that aside for a minute and just say, I've borrowed this task because I think it's a really excellent way to validate these models. So I'm going to come in and I'm going to compute the accuracy of classifying fine map EQTLs from a tougher negative set or sort of controlled for things like the distance to the transcription start site. And I'm going to do that with predictions derived from the previous Basenji 2 model and now from the new Informer model. And then each of the dots in the scatter plot is one of the GTEx tissues. So we do this whole training data random forest exercise for each GTEx tissue individually, thinking that they'll use different features, that the blood GTEx is going to use that PBMC cage, the liver GTEx is going to use hepatocyte samples. Here's the shape of the curve, and here is the accuracy broken down by different distances. We thought maybe we'd see that the Informer model was primarily boosting the accuracy at greater distances. While that's true, it's also true at all distances. So, by using long-range information, you seem to also be able to benefit what you're doing at shorter ranges as well. One final way we validated the predictions of the model is to make use of this wonderful data set produced by the CAGI competition, the most recent one, where there were a large number, I think 20-ish regulatory elements that were profiled in this saturation mutagenesis scheme with the measurements all made using a massively parallel reporter assay. So for these regulatory sequences, you're getting a measure of how expression is influenced for every single possible mutation across the sequence. And here you can see that data. Below that now, so again, this is very similar to what we do in silico. And so it's really wonderful validation data because we're often making these plots where we're measuring the influence of every single mutation. It's really important to be able to distinguish between the nucleotides that are participants and motifs that do matter from nucleotides that are just sort of flanking background, not as interesting. It's also quite important to get the sign right, which is a little more difficult than you might expect in our experience with some of these motifs. So this is a great data set to really be able to validate our models. And here you can see our track for a matched, this is the average across many cell types versus the gapped K-mer method, delta SVM, which is also a strong method for this task. And you can see here that although we're not perfect and then we're not getting every single motif perfectly, we are able to pick up on more than the previous best. And across all of the different regulatory elements, that's the x-axis here, all of these different gene promoters and or enhancers in some cases where they're underlined, we're able to boost the accuracy. You can look at it several ways. The competition held out some of the sequences to use as training data. And so in that case, we can come in with our high dimensional predictions and do a secondary regression to see what matters and what doesn't. And in that case, we were able to best the winners of the competition. However, we can also just come in and say, all right, well, each of these was done in a cell type. Let's just either pick the closest cell type or take an average across multiple cell types. And then there's no training. And that works surprisingly well too. I think that's the mode that we certainly aspire to, being able to pick out the exact cell type and be able to come in and make useful predictions for you. Okay, to summarize, in this work, we demonstrated that self-attention is a really powerful neural network block to incorporate for making use of long-range information, particularly enhancers, in making predictions about epigenetic state, transcriptional state. And that importantly has improved also our specificity across tissues and cell types. And as we move to a world where we have more single cell resolution experiments, and it's really the precise differences between these cell types and states that are the focus, that I think that'll be a quite important property to continue to improve upon. These models then, validated via two primary means, are able to better classify EQTLs and better match mutagenesis assays. So not only are we able to make good predictions for held out sequences, we're also able to make good predictions for genetic variants within those sequences. And you can check that out recently in Nature Methods here. Okay, just a quick where are we going next sort of roadmap. I think this work is far from done. It's way too much fun to stop now. One of the things we want to build upon is that even though we've improved the range and we're up to about 100 kb of being able to make a relevant prediction of a variant to gene expression, that doesn't quite cover it. There are known enhancers, there are distal variants that matter across distances that are even larger than that. So we want to be able to work with even larger sequences that will resolve this. This is a little tricky now because of transformer blocks. You might have noticed in the math, we have this attention matrix, which has quadratic memory in the length of the sequence. So if I double the sequence, I 4x the memory, and now I probably ran out of memory. And so these are the problems we're working on now, hopefully resolved with better hardware and perhaps better approximation algorithms in the software. Inspired by the work of Anshul and others, we are very interested in making predictions at finer resolution. A lot of these data are sort of older assays that maybe are okay to work with at 128 base pairs, but for a lot of the new data, there's major potential benefits to being able to work at finer resolution to really pinpoint transcription factor motifs. Of course, we again have this problem that if you decrease the bin size, you increase the sequence length, and you get the quadratic memory increase. So we'll be busy thinking about how to resolve that. And then, as I already mentioned, we're very, very interested in single cell genomics. The next talk is Han Yuan and my group with one idea we have about how to make use of that data. But we're sort of trying out lots of different ideas. And the simplest thing will just be to aggregate at cell type resolution and then continue as we're doing. But some of the assays like 3' single cell RNA-seq are a little bit tricky to work with. So a lot to do there. Okay, aspirationally added some slides on genome architecture. I've already talked about this, so I'm just going to cruise through them. We also have some ideas about how to work with Hi-C and how to predict Hi-C. This was published last year, so you may have already seen it. I thought it might be interesting to mention just because there's clear synergies between these two modeling approaches. If we had a better picture of the 3D genome architecture, we'd probably be able to make better gene expression predictions and maybe vice versa. So that's another thing we're thinking about a lot. And then broadly, this project with Hillary and Qingbo, we were using the scores to ultimately better fine map EQTLs, and that was very successful. But fine mapping GWAS is a different challenge. There's many differences in which features matter. There's differences in which genes are important and which are not. We've been thinking a lot about how to go from really high dimensional annotations to just resources that are more useful for the human genetics community, whether it's fitting classifiers to known disease variants or performing unsupervised dimension reduction. It's just a little hard to dump, 5,000 vector SNP vectors on people and expect them to make use of it. So we hope to help them figure out how to make use of it. And with that, I will thank Ziga and other collaborators at DeepMind, Vikram and Han, and my research group at Calico, Qingbo and Hillary at Harvard, genetics collaborators, and Jeffrey Freudenberg, formerly of UCSF, collaborator now at USC, starting his own group. And thank you to you all for your attention.
Thank you so much, David, for a great presentation. There is a number of questions in Q&A. We won't get through all of them, but they're ranked based on participant likes. So maybe I'll just ask you the first one or two, and then if you have a chance after we start the next session, you can continue to answer these through the Q&A window. Okay, so first question from Li Shen. A key feature of big NLP model is that you can incorporate a lot of unlabeled data. Have you explored this direction?
We have not. So my thought process here is that unlabeled data, a sea of A's, C's, G's, and T's, because of the very random evolutionary process, it's just not as informative. I think the protein field has a lot closer mapping to NLP, and that like not every amino acid sequence exists. Only some of them make meaningful proteins. And so we have not tried that. There's a few papers that have, and I think the results are not clearly beneficial. I think because we have so much labeled data, there's not as much motivation to work with unlabeled data.
Thank you. There's two other questions that are in computing like to each other that keep on getting swapped in terms of ordering. But so one of them is in terms of using your models in kind of a synthetic biology application to generate enhancer sequences. Have you explored that direction?
We have not, but other people have been really exploring with some really interesting approaches here. I mean, you can do really simple stuff, which is like pick a regulatory element that in the data has the specific properties you want, and you can mutate it and understand it. But then, yeah, I think optimizing those sequences, generating sequences that have certain properties is of great interest. And, because we've been thinking at really wide range, you're probably not generating like 100 kb, 200 kb blocks. You probably want to generate something smaller. And so I think here I'd probably go back to some of the older models like, the Basset, the Deep Seas, the BP nets, where you're working with a sequence that's more the size of the thing you want to create for your synthetic biology application. But it's a very interesting area of research.
Thanks. Okay, so the last question that we'll ask you live is, did the model learn about enhancer promoter distance rule? Have you looked into that? And are there specific distancing rule that the model has learned?
That's a great question. So yeah, we saw there were some recent experimental projects where people have actually done the experiment of, say, taking an enhancer and moving it at different distances. And it's been on our list to grab that data and basically do the same thing in silico and query the similarity of the results. In many cases, I think in this case I'm imagining, their CTCF and genome architecture is a huge component of their study. And so it would be a great benchmark, because it probably pushes the limits of what the model has really learned here. Broadly, the influence drops off with distance, but that's sort of averaged across everything. We haven't carefully done experiments like, here's a great enhancer. What happens if we move it all around? Does it matter if it's upstream or downstream? Does it matter if we flip it? There's, I think a lot of neat questions you could ask. Where I get stuck next is thinking like, that would be cool, I could write it up, and then some peer reviewer is going to tell me like, how do I know this is real? And I get stuck. So the validation is the missing piece for wanting to do a bigger study like that.
Thank you so much, David. I'll pass it over to Dr. Suen Lee, who's going to moderate the next session.
Great,
Okay.
Thank you.
Thank you for the great talk, David. So now we are going to hear 5 presentations from the authors of the papers selected for oral. So each talk and Q&A needs to be within 20 minutes, and then we will give a reminder to the speaker when the time is up, leaving a few minutes for questions. So if you have a question, please type in the chat box. If it's a short clarifying question, then I will try to ask the speaker to answer, but otherwise we'll wait until the end of the talk. So, and then these questions are ordered based on the number of likes. So please feel free to add your likes if you like those questions. So these remaining questions will be answered by the speakers after the talk. Okay, so the first talk will be will be about single cell Basset, the idea of using convolutional neural networks for modeling single cell taxic data. So the speaker, Han Yuan, is in David Kelly's group, and he's now in China. So we are gonna play the recorded talk. Okay.
I don't think we're hearing the sound, Suan.
I'm sorry. Anshu, what? You said something.
The sound doesn't seem to be coming through.
Really?
Yeah.
Oh my God. Okay. So let me then use my own speaker. Sorry. So let me try this way.
Hi everyone, my name is Han and I'm a computational biologist in David Kelly's group at Calico. Today I'll be talking about our model SC-Basset, which use sequence-based convolutional neural network to model single-cell ataxic data. Single-cell ataxic data is an assay that profiles accessible regions of the genome at a single-cell resolution. It is becoming more and more popular these days. It uncovers epigenetic landscape at a single-cell resolution and reveals cell-to-cell variation in chromatin accessibility and regulatory program. However, analysis of single-cell ataxic data remains challenging, mainly due to the high dimensionality and sparsity of the data. Typically, for each cell, at an accessible region, we usually have only 0, 1, or 2 reads. The goal of many single-cell ataxic modeling approaches is to overcome these challenges and to address the following objective. First, to characterize the cell-to-cell variation, to identify cell type cluster. Second, to infer the actual accessibility landscape at a single-cell resolution. And finally, to infer transfer factors that may be responsible for the variations in the chromatin accessibility. Many approaches have been proposed to model single-cell ataxic data. Here we're showing 2 examples of the state-of-the-art approaches in learning cell representation and denoising. The left one is systopic and the right one is scale. Systopic is based on latent diversity allocation or topic model, and scale is the variational autoencoder. Notice that both of these 2 models rely on peak-to-peak covariance to learn cell embedding and to denoise. Both of them ignore sequencing information. Since we know that transcription factors that bind ataxic peaks are sequence-specific, we expect that including sequencing information will help improve the modeling of single-cell ataxic data. There is a rich literature on predicting bulk epigenetic profiles from sequence information. And this line of research have already shown that sequences are very informative in terms of predicting accessibility. SC-Basset is an extension of such models, more specifically the Basset model from David Kelly to the single-cell dataset. Here's the SC-Basset architecture. So we start from a DNA sequence underlying a P, and we use one hot encoding to encode the DNA sequence to a 4 by number of sequences, number of base pair matrix. This input matrix goes through several convolutional blocks where each convolutional block contains the convolution layer, a pooling layer, and batch normalization layer. And the output is a latent peak representation. This latent peak representation finally goes through a linear transformation to predict accessibility of this peak of interest in each individual cell. So in this final linear transformation, the latent peak embedding is multiplied by this particular matrix of size number of cells times h. H is the size of the latent peak representation. So the columns of this matrix can basically be used as embeddings for each individual cell, as we will see later. The embeddings can be used for doing cell clustering, visualization, etc. The output of the SC-Basset model can be used as the noise profiles of each cell for accessibility of each cell. And we used a method called motif injection to infer transfer factor activity for any TF of interest with a motif. To benchmark SC-Basset, we look at 3 different data sets. The first data set is the effect-sorted hematopoietic differentiation data set. This data set provides ground truth cell type labels from flow cytometry. And the other 2 data sets are 10x multi-ohm data sets. We consider this multi-ohm data set to be valuable because they provide independent measurements of gene expression and chromatin accessibility in the same cell. As a first step, we want to ask whether SC-Basset can learn to predict accessibility at a single cell resolution with reasonable performance. So for the figure on the left, I'm showing the area under ROC curve for each of the holdout peak. You can see the mean performance is about 0.73, which is quite reasonable, although slightly worse than the performance in the bulk data. And the reason is because the SC-Basset, the single cell ataxic labels are noisy due to sparsity. So SC-Basset is already doing a pretty good job in terms of the prediction performance on holdout peaks. And on the right, I'm doing a sanity check for from another perspective. Because any cell-specific information is captured in the final fully connected layer, we expect the sequencing depth, which is cell-specific, to be captured by this last fully connected layer as well. Here we're plotting the sequencing depth in terms of log 10 UMI count versus the intercept of the final layer. And we see an almost perfect correlation. This indicates that SC-Basset captures the sequencing depth almost perfectly in the intercept of the final layer. So next, we want to evaluate the cell representations learned by SC-Basset. So as I mentioned before, we believe that weight matrix in the final linear transformation contains the implicit cell embeddings for each cell, and we use those to visualize and to perform cell clustering, etc. So this figure here is the t-SNE visualization of the cell embeddings learned from the SC-Basset model trained on this hematopoietic differentiation dataset, and the cells are colored by the cell types. These arrows vaguely shows the developmental trajectories that are corresponding to the ones here. Interestingly, we also see that the HSC populations are split into 2 groups. And this is due to a known batch effect that these 2 populations come from 2 different donors, which we'll talk a little bit later. To more quantitatively evaluate the cell embeddings we learned, we use 2 metrics, the adjusted rand index and the label score. For the adjusted rand index, it is basically we perform clustering our cell embedding and evaluate how well our clustering aligns with the ground truth. Since this metric relies on clustering and it is sensitive to hyperparameters such as hyperparameters and the randomness in the clustering algorithm. So we propose another alternative metric called label score. For the label score, we directly evaluate the nearest neighbor graph from the cell embedding. For each cell of interest, we look at is 10 nearest neighbor, 15 nearest neighbor, or 100 nearest neighbor, and ask what percentage of neighbors share the same label as the cell itself. So we can see across these different neighborhoods, SC-Basset outperforms all the other competitive methods, including cystopic and scale. As I mentioned a few slides ago, we see that in the hematopoietic differentiation dataset, the HSC cells are split into two groups due to batch effect. And indeed, these are the donors for these two groups of cells. So we wonder that we can adapt SC-Basset to account for known batch effect. And the solution we came up with is very straightforward. We added a second pass from the latent peak representation or the bottleneck layer to predict the batch-specific accessibility in each cell, given the dummy-coded batch information. The output of this pass is added to the original output and compare with the observed values. We expect that any batch-specific contribution to accessibility will be captured by this pass, whereas any biologically meaningful variation will be captured by the original pass. Indeed, when we look at figure B here, when we use this SC-Basset batch-corrected version, the two HSC population from different donors are now mixed together. Next, we evaluate the SC-Basset in terms of denoising performance. On the left, we sampled 200 cells and 500 peaks from the original cell by peak count matrix and show the heat map as well as the hierarchical clustering on the cells and peaks. We can see clearly here the cells are mostly clustered based on the sequencing depth, but not any biologically relevant information. Whereas on the right, we're showing the same heat map and for the denoised matrix. We can see that the cells of the same cell type share a similar accessibility profile and are clustered together. So this suggests that SC-Basset denoising can really uncover the biological signal in these accessibility profiles. Next, we evaluate SC-Basset in terms of TF activity prediction. So here we use a method called motif injection. Basically, for motif of interest, we feed into SC-Basset, a trained SC-Basset model, a pair of input sequences. One is a background sequence with dinucleotide shuffled peak. The other one is the same background sequence, but with a motif of interest in the center. So we can compare the prediction of SC-Basset on these two different sequences to evaluate the importance of this motif to each individual cell by looking at the change in predicted accessibility in each individual cell. So we benchmark the performance of TF activity inference against ChromeVar based on the consistency of inferred TF activity with TF expression from this multi-ohm dataset, which we have access to both the accessibility and the expression information in each individual cell. So for putative activators, these are basically the TFs that both SC-Basset and ChromeVar agree to have a positive correlation between expression and accessibility. For these putative activators, we can see that both SC-Basset predicted activity correlated much better with expression than ChromeVar. On the other hand, for putative repressors, so these are basically TFs that both SC-Basset and ChromeVar agreed on a negative correlation between expression and activity. For these TFs, we observed that SC-Basset predicted a much more negative correlation between accessibility and expression as compared to ChromeVar. So both of these two are statistically significant. As an example, here we look at PAX5, which is known to be a master regulator of the B cell development. So this cluster is the B cell cluster. And here we are looking at the expression of PAX5 on the UMAP. And we can see that it's specifically expressed in the B cell cluster. And so this figure shows the SC-Basset predicted PAX5 activity. And we can see that it is highly predicted to be more active in this B cell cluster and also Overall, across the atlas, the correlation between SC-Basset predicted activity and the expression is 0.32. Whereas for ChromeVar, globally, the correlation is only 0.09. And also, we don't see a high activity of PAX5 predicted by ChromeVar in this B cell cluster. Finally, I want to show an example where we can infer motif activity at a per cell, per nucleotide resolution using in silico saturation mutagenesis. In this example, we examine a known enhancer for the beta globin gene that regulates erythroid-specific beta globin expression. So this enhancer is located on chromosome 11. This is a 100 base pair window. By doing in silico saturation mutagenesis, or ISM, basically for each cell, for each base pair of the region, we mutate the nucleotide to each of the three alternatives and measure the average decrease in accessibility. This tells us how important the nucleotide is. The more important the nucleotide is, the accessibility will drop more once it's being mutated. We do this for all the cells, for all the base pairs in this 100 base pair window, and the figure shows the aggregate importance score for cells of the same cell type. For HSC, MPP, CMP, and MEP, these are the cell types in the erythroid lineage. We can see that the most important nucleotides matches the GATA1 motif and the KLF1 motif. So based on previous ChIP-seq study, we know that GATA1 and KLF1 actually regulate expression of beta globin by binding to this particular location. Interestingly, we also see that as the cells differentiate into the erythroid progenitor lineage, the GATA1 motif importance, the importance of this GATA1 motif also increase, as well as the KLF1 motif. On the other hand, for the cells that are not in the erythroid lineage, the importance for this GATA1 motif and KLF1 motif is no different from the background. In conclusion, we present SC-Basset, the sequence-based deep learning framework for modeling SC-ATAC data. It is trained to predict individual cell accessibility from DNA sequence underlying ATAC-seq peaks, and the learning a vector embedding to represent the single cells in the process. And we also show that a trained SC-Basset model can strengthen multiple lines of the ATAC single-cell ATAC-seq analysis. We demonstrate state-of-art performance on several tasks, including clustering, denoising, as well as TF activity inference. Finally, I'd like to thank my manager, David Kelly, and also additional members at Calico, including Vikram, Jacob, Mo, Nick, and Sarah, for helpful discussions. Apologize for not being able to answer the questions myself. David will help answering any questions you may have. Thank you.
Okay, thank you, Han. All right, so David has been very diligently answering all questions about his talk and then now, and then some of the questions on this talk. So now there's one question. I hope, David, are you around? Hope he can, yeah. Okay, so it's from Irene Kaplow. Can you explain how you define the peaks for the SC-Basset input and how you think this might affect your results? For example, if you cluster the cells initially and then call the peaks, SC-Basset might work better for cells in large clusters.
Yeah, this is a great question. So we stuck with what we'll call standard preprocessing, which is either the Cell Ranger pipeline in the case of data we downloaded from 10x, or a pipeline based on LSI and SVD methods, and peak calling strategies based on aggregating cells, calling peaks, and then in some cases, defining clusters, subsetting the cells, and a second stage of calling peaks. So this stage of the analysis is, I think, a little underexplored, and we're really interested to see if the sorts of machine learning sequence predictors might actually be helpful for the peak calling stage, because you really don't want to lose like a rare cell type peak call because you had to do this aggregating thing. But in everything you saw, we did sort of fairly basic work on peak calls that were available from those data sets. And then second part of the question, if we cluster the cells initially and then call peaks, that's effectively what's happening, which is a little unsatisfying because, you know, we're intrigued by the possibility that the Basset model here can help you cluster your cells. So if we require that you cluster your cells before you even run the method, that's a little unsatisfying, which is why I think it would be interesting to try to move a little earlier in the pipeline to the peak calling stage, but work in progress.
Okay, thank you so much, David. So it's already 10:21. There are two more questions. So I think David will need to type the question. So one of them is actually a really interesting question. Have you used explainable AI techniques such as deep lift or deep explainer, or maybe deep chef too? So David, please, if you are around, please type your answer. We need to quickly move to the next speaker. Thank you so much. So the next talk will be about interpretable deep learning methods for gene regulatory inference from single-cell multi-omic data. The speaker will be Manu Sarawat. Okay, you can take over, Manu.
Thanks a lot. Can you see my screen now? Yes. All right. Good morning, good afternoon, good evening, everyone. My name is Manu Saraswat, and I'm a PhD student with Moritz Mahl and Oliver Stegle in Heidelberg, Germany. And today I'll be talking about my work titled basically about gene regulatory inference from single-cell multi-omic data using interpretable deep learning. So transcription factors are proteins that can bind to cis-regulatory regions such as enhancers and promoters, and then control the expression of their downstream genes. And then some of those genes could themselves be transcription factors, and then could in turn affect expression of other genes. And together they might form networks, which we call gene regulatory networks. In recent years, single-cell methods such as single-cell RNA-seq and single-cell attack-seq have enabled the construction of gene regulatory networks at an unprecedented resolution. So we can combine RNA-seq with attack-seq together to construct these networks. There was a nice introduction about attack-seq data in the previous talk, but just to quickly go through it again. So the typical output from attack-seq is this peaks times cells matrix, which tells you whether a region in the genome is accessible in a cell or not. You could do further dimensionality reduction and clustering on these matrices to identify cell types. You could then look at differentially accessible regions in certain cell types, and then further go into which motifs are present in those differentially accessible regions to identify, say, master regulators in those cell types. Further, we can combine this data with single-cell RNA-seq data so that we can, say, correlate expression of a transcription factor with its accessibility, or further get enhancer to promoter links by correlating accessibility of regions with expression of genes. Now, to do these sort of analysis, typically we need different methods. For example, cystopic for clustering and getting set of differentially accessible regions, and Crombar for getting motifs in those regions. And also these methods are not designed to exploit the resolution of truly multi-omic data, which is both the measurements being performed from the same cell, and those sort of data sets are being generated more and more now. And these methods sometimes are also not scalable to millions and thousands of cells, which is like the scale of data sets being generated these days. And lastly, when we want to integrate data from many modalities, some of it might come up in the future, such as spatial transcriptomics data, these methods might fail. And to overcome these issues, deep learning methods can help us because they can integrate multiple modalities in a readily manner. For example, the most commonly used autoencoder architecture for single-cell RNA-seq or attack-seq or even joint analysis is shown here. So for example, if you want to integrate two modalities such as RNA and ATAC, you feed those two inputs into separate encoders and bring them down to a common latent space. Then they can be passed through a shared decoder to reconstruct the individual modalities. And then you can use this latent space for clustering and dimensionality reduction, so on and so forth. This is very nice, but the most commonly used methods, such as SCVI, do not incorporate sequence information into these methods. So you do not have information about cis regulatory elements. And also these methods have an inherent issue that they are not interpretable, that these latent dimensions might be hard to interpret. Like it's hard to make sense of what each of those dimensions represent, like which gene modules they represent or which peak modules they represent, for example. So what we want to do is that we want to combine principles of gene regulatory networks of transcription factor motifs affecting gene expression through cis regulatory elements. And we want to combine these principles with deep learning methods of autoencoders. And together we want to combine them to get interpretable latent variable models, which can leverage the multi-omic ATAC RNA data. And in this direction, we present our work, which we call SC-Dory, or single cell deep multi-omic regulatory inference. Now, at a broad overview, it's a topic modeling based approach. So it takes as input the peak accessibility profile of a single cell and the gene expression profile of the same cell as an input. And then we feed these profiles into an encoder to get a lower dimensional latent representations. Now it's a topic model, so we apply softmax to this layer. So we get a distribution over these latent dimensions or topics. Now, what we see is that these topics could represent, say, cell types, cell states, or just factors, if you want to say, if you're familiar with factor models, like decomposing expression and accessibility into just factors. Now, the difference here with existing autoencoder architectures is that instead of having a fully connected decoder, we use linear decoding using matrix multiplication. For example, to reconstruct the peak accessibility profile, we break down the decoder into 3 matrices, each of which represent some biological phenomenon. And similarly, we do the same for reconstructing gene expression profile as well. Now what I will do is I will go through each of these module one by one. Module 1 for the encoder, module 2 for peak reconstruction, and module 3 for gene expression reconstruction one by one, and then explain what is going on in these modules. So the first module is simple. It's like any existing encoder that it takes as input the peak accessibility profile and the gene expression profile as input, and gives you a distribution over these latent topics. Now these topics could be further used for dimensionality reduction, clustering, and visualization. And as I will talk about later in results, that some of these latent dimensions or topics represent cell types. Now let's talk about the reconstruction of peak profiles or the accessibility profile. Now to do so, we decompose this peak accessibility profile into 3 matrices. First is the cells times topic matrix, which is coming from the encoder. Then what we do is we learn an embedding matrix for the peaks. So what we do is for each peak, we obtain an embedding using a sequence-based convolutional neural network. And instead of using fully multi-layer CNN such as Basset, what we do is we use linear convolutional neural networks, which basically give the embedding in the form of transcription factor motifs. So each of those dimensions in this embedding represents a transcription factor motif. So we keep this, we learn this matrix in the first step using a CNN, and we keep it fixed. Then we learn an embedding for each topic in the same embedding space. So over here, each row represents a topic, and the model will learn the embedding for each topic in the same embedding space. So if you then look at this dimension for this particular topic, it will tell you how important this motif is for that topic. And then when you take the product of these 3 matrices, cells times topics, topics times embedding, embeddings times peaks, you get the cells times peaks profile, which is the reconstruction for the accessibility. Now, once the model is trained, if you take the product of these 2 matrices, this is the topics times peaks matrix, it essentially gives you the important modules for each important modules of peaks, or a set of co-accessible peaks for each topic, which can be used for further interpretability, and I will talk about that later. And also each topic has an embedding, as I talked about, in terms of transcription factor motifs. So it tells you what motifs are important for that topic or that cell type. Let's talk about the gene expression reconstruction. These three matrices remain the same, that we have a cells times topics matrix, we learn a topic embedding matrix, and this matrix it's fixed, it's learned from the CNN. Additionally, what we do is we learn a matrix of peaks times genes, which could represent putative enhancer promoter linkages. In simple words, what we are trying to do here is we are trying to learn the expression of a gene using the set of peaks which are co-accessible with that gene. So the embedding of each gene is basically a weighted sum of the peaks which are co-accessible with that gene. And when you take the product of these four matrices, you get a cells times gene matrix, which is the reconstruction. And then you can put that into the autoencoder loss. Again, like peaks, if you take the product of these three matrices, you get a topics times genes matrix, which is essentially giving you important genes for each topic or like set of co-expressed genes or gene modules, which could be used for further interpretability. Additionally, if you look at the embedding of genes, so this is genes time embedding matrix. And as I said, each of these embedding dimensions represent a transcription factor motif. This could further give you insights into gene regulatory networks, basically motifs which are present in the putative enhancers of that gene. Finally, the bit I was talking about for generating embeddings of peaks. So we use these convolutional iterative models, which I presented in the Mlcb conference last year. So they are a linear form of multi-layer CNNs. So they consist of these convolutional units, which is one single convolutional filter followed by two fully connected layers. And then you have an output from this unit. And to compute the final prediction of the model on a one-hot encoded DNA sequence, you just take a weighted sum from these many such units. And once this model is trained, you could go and look at these filters, convert them into PWMs and see if they correspond to binding motifs of some transcription factors. So in the first step, what we do is we do prediction of accessibility from a sequence in a cell type specific manner in a supervised way. So we assume that we have cell type labels for our data, and then we use this model to generate embeddings for each of the sequence. And then once these model is trained, we just keep this purple matrix fixed based on these outputs. So this is the data set I'm going to be showing the results of the modeling approach. So it's a multi-omic ATAC RNA data set from the same cell of mouse gastrulation. Consists of 30,000 cells sampled across 3 time points. Over here, I'm showing the visualization of RNA and ATAC modalities separately. They are both measured from the same cell, but just to show that there is concordance between the type of cells we are getting. You can see different germ layers, erythroid and mesoderm, ectoderm, and so on. So firstly, what we do is we look at the topic distribution for the different cells, and we can visualize them on a UMAP. And this is what it looks like. So we can see that different germ layers are very nicely separated when you visualize these on the UMAP. Then additionally, what we see is that certain topics or latent dimensions correspond to fixed cell types. For example, topic number 0 is highly enriched in brain-related cell types. Topic number 5 is enriched in mesodermal and specifically cardiomyocytes. And topic 7 is more enriched in erythroid lineage. So we see that we are recovering cell type specific topics. Now what I want to talk about is how we can interpret these topics identified by SC-Dory. So we see that some of the topics are cell type specific. I want to show how can we make more sense of them biologically based on how we have developed the method. So first way is using the gene modules. So we can find set of co-expressed genes for each topic. Second is peak modules. We can find a set of co-accessible peaks for each topic. And thirdly, we can look at the group of TF motifs which are active in each topic. So talking about the first approach, which is the gene modules, essentially we take the topics times gene matrix, which is the product of these 3 matrices. And then, for example, I'm showing you results on this cardiomyocyte specific topics. So we take the genes which are important for this topic and do some sort of gene set enrichment. And we see that it's enriched in terms such as heart development which makes sense because these genes are important for this topic. Similarly, we can also look at the topics times peak matrix, which will give us a set of peaks which are co-accessible in that topic. And then we can do further enrichment on those peaks. Over here, I'm showing enrichment of known transcription factor binding sites from public ChIP-seq data on the peaks which are important for this topic of cardiomyocyte. And we see that it's enriched in some of these transcription factors which are known regulators in cardiomyocytes. And this enrichment was done using Unibind enrichment tool. You could also look at, you could also assign genes to these peaks. So like the nearest gene or something like that, and do some sort of enrichment on the gene as well to get more insights into what sort of molecular processes are being controlled by these set of peaks. And thirdly, for each topic, we can also look at what transcription factors are important. And to do so, we just have to look at the embedding of that topic. So each of these dimensions represent a transcription factor motif. And for the cardiomyocyte specific topic, we see which of those embeddings have the highest weight. And one of the highest weighted embedding dimension was for the GATA motif, which is a known regulator in cardiomyocytes. And then further, we can visualize these embeddings on a UMAP at a single cell level also, as I've done here. Now, there's a bunch of other tasks we can do with this model. What I've shown is just like this, like scratching the surface. I just want to quickly show like some of the ongoing work, which is basically we can have a common embedding of cells and genes together. So if you look at these two products, which is cell-time embedding matrix, and this is a gene embedding matrix. Now what we can do is we can visualize genes and cells together on one UMAP. And the idea is that genes which are cell type specific, we expect them to be clustered together with the cells of that cell type. So over here in this UMAP, I'm showing you the orange colored cells are from cardiomyocytes, and I'm showing some of the known marker genes of cardiomyocytes which are clustered at the same position in the same region. Similarly for erythroid, important factors such as RUNX and ENK1 are like in the erythroid region, clustered with the same cells. And similarly for neural crest as well, SOX10 and TFAP2B transcription factors are clustered together with neural crest cells. And so what we can do, this approach, we can use it to identify marker genes. So we can compute the similarity of embeddings between genes and cells. And the genes which are more similar to cells are potentially marker genes or even master regulators if those genes are transcription factors. So in conclusion, SC-Dory is a deep learning framework which incorporates principles from GRNs to obtain interpretable latent space from single-cell multi-omic ATAC RNA data. The linear decoder network provides biological interpretation to gene regulatory dynamics using topics times gene matrix, topics times peak matrix, and topic embedding matrix. With that, I will conclude my talk, and I'm happy to take questions, comments, and suggestions, even like discussions later on. What more we can do with such a modeling approach. As I said, I'm not talking about everything which can be done, only some of the key points which we have explored until now. Thank you.
Okay, thank you so much, Manu. So I see your question in the chat box. From Irene Kaplow, you described how SC-Dory enabled you to detect known biology. Have you have any potential new biological findings from this method? So if so, how do you suggest following up on that?
Yeah, I mean, for now, we wanted to first validate like the things we get from the model make sense. So first step is to validate from the known biology. We do have some interesting insights, for example, from the enhancer promoter matrix. We get that certain peaks are have high weight for certain genes, so they could be acting as enhancers. So we could maybe follow up on some of those findings of potential enhancer promoter links.
Okay, thank you so much. There's one more question. So have you tried using more complicated CNN architectures to better capture cell state specific transcription factor motifs?
Yeah, that's a good question. So yes, we have tried. The problem with the complicated architectures is that you lose the interpretability. So what I tried was embeddings from SC-Basset. So I use the same approach, SC-Basset predicting single cell accessibility and then using the final layer embeddings in the model. So in terms of the reconstruction performance we get, it improves slightly with such a complicated CNN, but then you lose the interpretability. So in the case I presented right now, each of the dimensions represent a motif. You don't get such interpretability anymore. But we could think of ways to basically get best of both worlds, and which is something I'm exploring right now. How good is the reconstruction quality of such a linear encoder? It's not as good as what you would expect from a nonlinear decoder, but it's not very bad. And there have been studies like this is not the first approach to try linear decoders. There are other papers already doing so. And so they have compared extensively the performance of such methods. So it's slightly less, but you get the interpretability advantages, which is what we are more looking for here.
Okay, sounds great. Okay, thank you so much, Manu.
Thanks a lot.
Okay, so next presentation is about SC-Origami for predicting 3D chromatin structure in cis-regulatory interaction networks from single-cell chromatin accessibility data. From Vian.
Yep, thank you. Okay. Hi, everyone. My name is Vian, and I'm a PhD student from Christina Leslie's lab at Sloan Kettering. So today I'll be presenting a work in progress model called single-cell origami, and we are predicting the 3D chromatin structure from single-cell chromatin accessibility data. And before moving on, I just wanted to acknowledge everyone on our team, Ray, Arnav, and Alireza, as well as our PI, Christina, Jeff, and Bill, who has offered incredible amount of support. Okay, so first, as a quick background, I'm sure you're convinced by the previous talks already that the 3D genome organization plays an important role in gene regulation. So you can imagine when the chromatin folds, as shown on the figures on the right, it can bring the trait elements closer together in the 3D space and hence regulate the transcription of the downstream genes. And in order to better study and identify these regulatory interactions and be able to map which regulatory elements regulate which downstream genes, high throughput methods such as Hi-C and Hi-Chip can be conducted in order to capture these events. And so from a Hi-C experiment, you will get something called a contact map that looks like the matrix on the right. So in this matrix, you see that the red spots in the matrix indicate that the corresponding genomic regions are in close proximity with each other and hence likely to be in contact. So while these assays are powerful methods to identify regulatory interactions, there are some drawbacks, mainly that they're difficult and costly in order to obtain a high quality contact map, and especially for populations that are small or difficult to isolate. And to this end, a lot of computation methods have been proposed and have succeeded at predicting these chromatin interactions or contact maps from other modalities that are relatively easier to obtain. For example, DNA sequences and from epigenetic marks. While these marks are powerful, there are some drawbacks related to each. For example, using DNA sequences, it's relatively more difficult to obtain a cell type specific prediction because the underlying sequences across the cell types remain the same. And on the other hand, while using epigenetic marks can give you a cell type specific prediction, it's not always the case that you have the full set of marks needed for the model to predict in your cell type of interest. And to this end, we propose single-cell origami, which predicts chromatin interaction from single-cell chromatin accessibility data, which is becoming increasingly accessible and available thanks to the commercialized kits. So specifically, we're using single-cell attack-seq data, which is an assay that captures chromatin accessibility at a single-cell resolution. So if we take a step back and think about what types of information we can extract and harness from single-cell attack-seq data that can help us to predict chromatin interaction. There are two things. The first is the aggregated accessibility across a population of interest. So what you get is this 1D track of accessibility aggregated across a cell population. And this is useful for predicting interaction because we know that chromatin accessibility is a hallmark of active regulatory elements. And secondly, another useful input is the co-accessibility matrix. So what the co-accessibility matrix captures is how correlated the accessibility are for pairs of genomic regions. So essentially, it's capturing the degree to which pairs of genomic regions become accessible and inaccessible simultaneously. And indeed, in a previous study that proposed the method Cicero, the authors found that, so Cicero, as a quick background, is an unsupervised method that tries to infer chromatin interaction from this co-accessibility matrix. And so what the authors have found is that pairs of genomic regions that are found to have a high co-accessibility are actually enriched for regulatory interactions. So we thought these two information are both useful in order for our model to predict chromatin interaction. And so using these two inputs, what we're trying to predict is a Hi-C DC plus normalized Hi-C contact map. And for each input region, for each 3 megabase input region, instead of predicting the whole contact map, what we're trying to do is simplify things and only predict the interaction between the genomic region in the center and its neighboring regions within a 1 megabase distance. Okay, so since the region that we're trying to predict is within this black box V-shaped stripe, we can also simplify our co-accessibility input. So instead of inputting the full co-accessibility matrix, we extract the region that is relevant to the interactions that we're trying to predict with some context to reduce some overhead. Okay, so now that we have seen the input and the outputs of the model, we dive a little bit deeper into the model itself. So single-cell origami is a simple convolutional neural network-based model that has 2 feature extractors. The first one is for the aggregated accessibility track, as shown here. And it first consists of a series of 1D convolutional blocks, followed by an outer sum operation that is inspired by the Akita model. And the idea of this outer sum operation, which is detailed here, is that we want to aggregate the features that are extracted from pairs of genomic bins, and then make the prediction about their interaction. And afterwards, we perform 2D convolution on the product of the outer sum. And specifically, we also add some skip connections in order to preserve some of the higher level information. Okay, now the second feature extractor is simple for the co-accessibility stripes that we have extracted from the matrix. So it's a series of 1D convolutional layers, followed by a dense layer. And then finally, we concatenate the features that are extracted from the 2 inputs, feed them into the final dense layer, and predict the V-shaped stripe that we described in the previous slide. So this model is trained end-to-end on the GM12878 cell line data, where we have a high resolution, Hi-C data available. And the training, validation, and testing are split by chromosome, as shown here. And specifically, we optimize the mean square error loss. So after predicting the V-shaped stripes, we can combine them into a full chromatin contact matrix similar to that generated by the Hi-C experiment. Okay, so after training, we want to evaluate how well the model performs on the held out chromosomes in GM12878. First of all, we want to visualize our predicted contact map. On the left, we show the predicted contact map on the top, as well as the ground truth. On the right, we have some more quantitative assessments of how well our models capture the significant interactions, which are the top 5% bins, top 5% significant interactions. and also the Pearson correlation between the predicted and the ground truth high-C contact map. We also performed ablation tests to see the contribution of each input on the prediction of the interactions. And we saw that on the second row in the contact map, when we ablate the co-accessibility, we're still able to capture the major interaction structures. However, the resolution of such prediction is much worse compared to when we have the co-accessibility input. And secondly, if we ablate the accessibility information and only use co-accessibility, there's a lot of long-range interactions that are missed. And similarly, quantitatively, we see that when we use both the aggregated accessibility and the co-accessibility as the input, the model performs the best, both in terms of significant interactions and Pearson correlation. And also we see that all models outperform the average activity baseline, which is just predicting the mean of the training data. So after evaluating on GM12878, we wanted to see whether we can apply this model, the single-cell origami model that is trained on GM12878, where there is high-resolution, high-C data available, to a new cell type. However, there are some problems associated with this transfer, mainly because there are distribution shifts between our training cell types and our test cell types. That might be caused by there being different numbers of cells, or there being different qualities in the input data. So in order to address this problem and improve generalizability, we borrow some ideas with data augmentation from the computer vision world. So imagine when we are doing image classification and you have a cat image, what people do is that we can inject noise, rotate, or crop this image on the left. And we want to train a model to learn that despite all of these things that are done to the image, it's still a cat. Similarly, in our case, what we do is during training, we randomly subsample different numbers of cells and help the model to learn that despite there being different numbers of cells sampled, hence there being different levels of noise and variability in the training data, the same contact map should be predicted because these are cells that are sampled from the same cell type. Indeed, we see that when we perform the subsampling data augmentation approach during training our single-cell origami model, the generalizability is significantly improved when we try to apply the model on the new cell type. So here I'm showing the results for when we train on GM12878 data and test cross-species and cross-cell type on mouse germinal center B cells. So we see that both the significant interaction identification as well as the Pearson correlation are significantly improved when we perform subsampling compared to when we don't. And it's also worth noting that when we do the model subsampling perform comparably to the model when we evaluate on the held chromosomes on the training cell type. And finally, we can visualize some predictions at some genes of interest. So for example, PAX5, you've seen this gene 2 talks ago. It's an essential transcription factor for regulating B cell development. And we see that in the germinal center B cells, if you look at the ground truth high-C contact map, there are some strong interaction structures that are found and is indeed captured in our predicted contact map. However, when we apply the model to Tregs, we see that this interaction structure is not identified and it's also missing in the ground truth data. And similarly, we can look at another gene, which is FOXP3, which is an essential transcription factor for regulating Treg development. And this time we see that there are strong interactions around the FOXP3 locus in the Tregs, as is predicted. However, if you look at germinal center B cells, the interaction is much weaker and we don't predict that interaction. And so the takeaway of these two visualizations is that single cell origami seems to be able to make cell type specific prediction at genes of interest. And so in conclusion, we've shown that single cell origami is able to predict cell type specific chromatin interaction events from single cell ataxic data. And we also saw that both the aggregated accessibility and the co-accessibility information contribute to predicting the chromatin interaction. And it seems like the aggregated accessibility a little bit more than the co-accessibility. And finally, we saw that by performing subsampling during the single cell training, we greatly improved the model's generalizability to new data sets. And so since I mentioned that this is still a work in progress, there is a lot that needs to be done. So the first few definitely involves further evaluating the quality of our predicted high C contact map, especially to see if we can identify real task structures and how they compare to the ground truth. And also we definitely need to benchmark our model, compare with some of the other models that have been proposed to see where we fail, where do we do better. And then definitely finally, the most interesting part is to do some saliency analysis to interpret what the model has learned from the single cell ataxic data, such that it's able to predict these chromatin interactions. And with that, I want to thank everyone from my lab, Christina, Ray, Alireza, and Wilfred and Merve, who helped me a lot in processing the high C data. Also Jeff, Bill, and Arnav, as well as collaborators from the Melnick lab for sharing the Germinal Center B-cell data. And happy to answer any questions.
Thank you so much. Thank you for the nice talk. So I see one question from Irene Kepler. So I think that GM12878 and mouse germinal center B cells are both blood cell types. Did you try SC-Origami trained on the GM12878 to a non-blood cell types? If so, how well did that work?
I see. So far, what we have tried is to apply the model on several other immune cell types. So naive CD8 B cells, Tregs, effector CD8 B cells. Yeah, it's an interesting point. We'll definitely try on more cell types and see what happens.
Okay, so David Kelly asked the question. So have you considered any strategies to help the model figure out which accessibility sites have CTCF binding?
So just to clarify, you mean adding additional input information during training, such as like adding DNA sequences or maybe ChIP-seq data for CTCF to help the model predict better?
Yeah, I think that's what he meant. So let me read again. Yeah.
Oh, yeah.
Yeah, so any strategies to help the model figure out which accessibility sites have a CTCF binding?
I see. Yeah, I think that would be a very fruitful effort. We've definitely considered adding maybe DNA sequences as an additional input or adding additional epigenetic tracks. Actually, there is another model from our lab that is soon to be submitted called Epiphany. And there they use a bunch of epigenetic marks, including CTCF, to predict the high C contact map. And so in this model, I think we can either add a CTCF track as an input, or we can do some sort of pre-training to train to predict the CTCF ChIP-seq track from single cell ataxic data, and then use that pre-trained feature extractor as part of the single cell origami model. I think that's also an approach that we can take.
Okay, so Bill Noble asked the question. So you described the average activity baseline as being the average of the training set. Can you describe it further? My intuition is that it should have been the average per pair signal across the other cell types in the same chromosomes as your test set.
I see. So currently what we're comparing in the average activity is we're trying to see, okay, what happens if the model didn't really learn anything and it's just trying to predict the mean of what it's trained on. So we haven't tried to benchmark against when we compare, when we just predict the average of all of the test set. Currently, we're training on one cell type and applying to multiple other cell types. So that's something that we can definitely try, but we haven't done so far yet.
Okay. All right. So last question, Oriole. From accessibility data, could you model? Could the model predict the impact of the lesions that would disrupt TED boundaries?
Impact of the lesions. Good question. I think that will be something fun to explore when we actually do some these in silico perturbation to see if we mask certain peaks or certain regions, whether that affects the accessibility. But so far we haven't done any perturbation analysis yet. I think that will be something interesting to explore, like maybe mapping the having the CDCF track and then mapping where the accessibility regions are and see what happens if we mask certain regions of the accessibility.
Okay, thank you so much. Thank you so much for the talk and then all these answers for the questions. So next we will go to oral number 4, titled Towards Data-Driven Design of Context-Specific Regulatory Elements. So Walter is here as a speaker.
Great, thank you very much. Yeah, so today I'm excited to present our work we did regarding data-driven design of synthetic sequence elements, in particular, sequences that are predicted to be active in a specific cellular context of interest. This is work done with Peter Bromley, an extremely talented and determined individual. It's an ongoing project, and so we're sharing this in the hope that it can be useful for others, and we would love to collaborate. We want to particularly thank the 4 anonymous reviewers of this work who have actually provided extremely useful and encouraging comments to this work. Sometimes it's very nice to see positively slanted and helpful reviewer comments every once in a while. It was really great. Thank you for that. Okay, so if we look at regulation of gene expression, then we see that genes are regulated by regulatory elements in a way that is at least in part encoded in their genomic sequence. This means that, at least in principle, it's possible to do cellular programming by manipulating or perturbing these kind of sequence elements, but the rules for doing so are largely unknown. There have been a number of experimental efforts to do this at various scales, but those are often guided by sort of preconceived notions and assumptions about what actually constitutes or makes up a regulatory element. This has so far hampered progress in terms of designing synthetic regulatory elements, for instance, for applications in gene therapy. So as we saw in the previous talk, one of the hallmarks of regulatory elements is their accessibility to DNA binding proteins, such as transcription factors. And so we have previously in other work collected and analyzed chromatin accessibility data across more than 700 different biosamples in human. And so today we are talking about work where we use these data to train generative and discriminative models for synthetic sequences. As I mentioned, with the particular goal of evolving these in silico towards a number of predefined cellular contexts. This is definitely building upon and inspired by other work from others that came before us that have used these kind of large-scale data-driven approaches to design and perturb sequences for various application areas listed here. So looking at DNA binding proteins and how they bind to accessible chromatin is a useful way to identify regulatory elements. And this accessible part of the genome can be mapped using DNase I digestion. An alternative way of doing so would be, for instance, ATAC-seq, as we've seen previously. We use DNase I as it's sort of the gold standard for these type of approaches. Briefly, how it works is DNase I is an endonuclease, which basically means that whenever DNase I enzyme observes any kind of accessible DNA, it'll cleave it, it'll cut it, ending up with a large number of these kind of DNase I fragments, which we subject to paired end sequencing, and we map the 5' ends to the genome. And that's what this looks like here for a small portion of the genome for an erythroid cell type. we see that there are these local regions of enrichment of reads indicating that those areas are accessible versus the ones that are not. As I mentioned, we've done this across more than 700 different human biosamples, representing more than 400 different cell types and states. I'm showing you here a very small subset of the hematopoietic system, basically to show you that there's a huge diversity in terms of occurrence patterns across these different cell types. Unlike genes, for which we have these very nicely curated annotations like GenCode and RefSeq, we don't have these for regulatory elements or for accessible elements. And so we set out last year to do so, to build essentially a common coordinate system of accessible genomic regions. I'm mentioning this because it's the data we're using for today's modeling, but also because I think it's a really useful general data set for the machine learning community, because we don't just annotate these putative regulatory elements based on the start and end coordinates, but we also give an estimate based on their centroid position, based on essentially peak summit estimates across all samples, as well as a measure of sort of the positional stability of these things across cell types. So we have 3.5 million of them, and we are able to annotate all of these for the first time, basically based on their accessibility profiles across cell types. So we were essentially doing a matrix factorization of this large accessibility matrix, which allows us to annotate each of these DHSs with one or more of these DHS components that each reflect a different cellular context. So you have cardiac, neural, stromal, etc. We sort of validate these kind of annotations by looking at what the primary transcription factor motifs are that are enriched in these kind of DHSs, indeed confirming in most cases very well-known factors involved in these kind of cellular contexts. So those are the data that we use for our setup. Our modeling setup is relatively straightforward one, where we essentially use adversarial training to take a random input seed, push it through a generator model that produces a 200 base pair sequencing. We train this generator using our real DHS sequences, so DNase I hypersensitive site sequences. And we have a separate discriminator that basically determines whether a sequence was generated by the generator or actually came from the real set of sequences. So during training, both the generator and discriminator become better and better at what they're doing, with the end result of having a generator that generates sequences that presumably look like DHSs. So we have a number of steps to validate that this is indeed the case. Simplest of those is look at GC content of these sequences, which is very much in line with the training set. But more interesting, we can look at different values of k-mers. So in this case, we have essentially for all possible 4-mers, we're looking at the distance to the nearest neighbor within the training set, for instance here, as well as between the generator output and the training set. This confirms that our generator is relatively stable, doesn't really suffer from mode collapse. At the same time, all the generated sequences, if we blast them against the human genome, they don't meaningfully map to the human genome, indicating that the model also doesn't just sort of memorize sequences. Now that we have this 4-mer space, this k-mer space, we can basically project all 750,000 training sequences into this 2D UMAP space and color them, in this case, according to GC content, indeed showing that there's a large separation based on GC content, with high GC content in the lower right and low GC content in the upper left. Assuringly, when we now generate 1,000 synthetic sequences, we see two things. One, that there's indeed, again, a separation based on GC content, but maybe more importantly, that all these sampled points are nicely within the confines of the 2D UMAP of the training data. So this kind of confirms that these sequences really do look like DHSs, but of course it's unclear in which cellular context they would potentially actually be accessible. And so to sort of further enforce this, we add a separate classifier model to our setup, which allows us to essentially iteratively tune any of these generated sequences by iteratively adjusting the seed value, pushing it through the generator, and then having a classifier determine whether, what DHS component best corresponds to that sequence. So we build a classifier based on these 16 DHS components, so lymphoid, neural, stromal, etc. And then we use a large number of high confidence DHS sequences of which we're pretty certain what their cellular context is. So they're lymphoid or myeloid or neural, etc. And then we tune these sequences iteratively by adapting the seeds. This classifier performs reasonably well. We see that it nicely generalizes to an independent test set. And we see that there is a reasonable range of F1 scores, considering that this is essentially a multi-class classification. Interestingly, if we look at the filters of this classifier, which is a convolutional neural network, we see that there are many filters that are sort of, at least in part, reflective of a lot of these previously characterized motifs associated with these DHS components, which I showed in the third slide or so, which is encouraging that this classifier is indeed picking up on relevant sequence signal. So now what we can do is we can say, let's start off with, again, the backdrop of the training set, and then generate 1,000 synthetic sequences colored according to what the classifier initially thinks these sequences represent. And now through a number of iterations, we can start tuning these in silico towards, in this case, the lymphoid context. So you see that within not too many iterations, the fraction of sequences that is classified as lymphoid it becomes quite large. The interesting thing is that we can do this not just for a single context, but we can start off with the exact same set of generated sequences and basically in parallel evolve these in silico across towards all these 16 components. And again, relatively quickly, it should become clear that what that context of interest is for each of these 16 according to the different colors. Important to mention here is that once again, even during the tuning process, we see that these points do not stray outside of the backdrop of the training sequences. Indeed, if we look at stability of training, we see that across even thousands of iterations, we see that the GC content stays relatively stable. And if we look at, again, the same K-mer analysis, we see even then that there's, things look relatively stable up until some point where we start seeing some slight deviation. But then we're looking at 10,000 iterations, which is typically not as far as we go. We also see that the tuning process is relatively stable. It takes on average about 3 iterations to only make a single base pair change. And even after 10,000 iterations, we see that the majority of the sequence is typically still the same as the starting sequence. What's interesting to see is that as the tuning loss goes down over a number of tuning iterations, the number of sequences, the fraction of sequences that has a relevant motif for that particular DHS component rapidly increases. And this kind of raises the question on, where should you stop tuning? Should you just keep going, or should you stop at some point? And this is something we're currently exploring, and it's a bit of a philosophical issue here, right? So we had a number of considerations here that might be useful for folks attending this meeting. One is to look at the sequence characteristics, and in particular, something known as Shargov's second parity rule. So Shargov's first parity rule is a simple one, and one that everybody here should be very intuitive and familiar with. It basically states that for any double-stranded DNA molecule, the number of A's is equal to the number of T's, and the number of C's is equal to the number of G's, which is, for all of us, very intuitive because it's essentially Watson and Crick base pairing. What's maybe less intuitive is the second parity rule, which states that this is essentially also somewhat the case for even single-stranded DNA molecules. And exactly why this is not entirely true, but it is a well-established notion that this is actually true at a population level. So we were looking into sort of using this at the very least as like a soft constraint for early stopping. However, then we started looking at a number of these real DHS sequences, and these easily violate any of these and a number of other rules that people have applied for sort of penalizing synthetic sequences, like large numbers of repeats and things like that. And so, even though these kind of sequence characteristic metrics have definitely potential value at a population level, we think they're too unstable for individual sequences. Instead, what we're looking at now is basically tuning performance. So what is the earliest point where a classifier becomes and remains 100% committed to a chosen cellular context? So if you're evolving towards a lymphoid component, at what point is the classifier sure it's a lymphoid sequence and then never changes its opinion again? And we're looking at various ways of maybe combining this with speed of convergence, or whether there's certain motifs picked up and things like that. Like I said, it's a little bit of a philosophical issue here. And so really the proof of the sequence is in the assay. And so we're developing a number of assays, undertaking a number of assays now to test these kind of predictions. There's various options. Two I want to highlight is an episomal reporter expression assay, such as MPRAs, where you would basically, look at the ability of your synthetic sequence to drive expression of a particular reporter. This is well established, and it gives you an immediately functionally relevant readout, in this case, expression. Alternatively, we are also looking at actually integrating these sequences in genomic loci of choice, which will give you actually a readout in the genomic context. And you could even look at sort of a more direct readout of what you're trying to predict in your model, namely DNA accessibility. All of the software and models are available here, and implore you to check it out. This is mostly work, as I mentioned, by Peter Bromley, and I want to thank the other folks in my group as well as collaborators, Georg Selig's group, who will be speaking tomorrow. And these are names of some of the folks who've moved on to bigger and better things. This could also involve you. We are looking for postdocs, students, and other folks. This type of work is something you're interested in if you care about regulatory genomics and large-scale analyses and data visualization, as probably a lot of people here. Please get in touch. We can be very flexible about any kind of working arrangements. Thank you.
Okay, thank you so much for the nice talk. So I see a question by David Kelly. Is it repetitiveness? For example, tandem repeats or many copies of the same motif. Is it an undesirable property for your generated sequences?
Yeah, so this is something, so that's an interesting question. So this really kind of depends on, so we are bounded by whatever the generator produces, right? And so in terms of many copies of the same motif, like you might say, like, well, if you want to have, if you want to have a sequence that really does very well for your classifier, you just plug in a sequence with 6 of the same motifs or something like that. It's bound to do well. The thing is that the generator is not going to produce these type of sequences because they don't actually typically occur in real DHSs, right? And so in that sense, it's an undesirable property, perhaps not because of the classifier, but it certainly is because of the generator, right? So we really use this generator model as sort of a general oracle of what a DHS should look like, regardless of which cellular context, and that sort of really bounds the properties of that, basically.
Mm-hmm. Okay, thank you so much. So the next question is from Bernardo Armaida. So how diverse were the generated sequences for a given cell type, for example, related to diverse motif combinations?
Yeah, so we have not looked at this in terms of diverse motif combinations. We have looked at this in terms of general diversity in K-mer space, and we see that diversity goes down a little bit as you tune these sequences, as expected, because you are essentially, in case of the UMAP plot, you are sort of nudging them in a particular direction of that K-mer space. So you will see that diversity within that set in general goes down a little bit. In terms of diverse motif combinations, we have not looked at that yet beyond simply looking at sort of the number of motifs that's being picked up per sequence, which is typically very low, like 0, 1, or 2.
Okay, so last question from Irene Kaplow. Are your generated sequences more similar to sequences in stronger DNA's peaks than to sequences in weaker DNA's peaks? So do they tend to be more similar to sequences in cell type specific DNA sites than to sequences in DNA sites in most cell types?
So that is an excellent question. So we are, as you saw, we are looking at 750,000 DHS sequences, which is a small subset of the 3.5 million that we have total, which means that we are already enriching for the more sort of the more higher confidence or the stronger DNA's peaks, just to make sure we have less false positives in our training data. But we haven't looked at whether the generated sequences going back and whether they are actually more similar to weaker DNA's peaks or stronger DNA's peaks. In terms of the second part of the question, do they tend to be more similar to sequences in cell type specific DNA sites? Once you are tuning these sequences towards a particular cellular context, you would expect that to be the case, right? Because the one thing I didn't show is, like, as you are tuning these sequences to a particular cellular context, you start picking up more, the sequences start picking up more context relevant motifs, but only for that particular context, right? And so given how cell type specific DHSs are enriched in these type of motifs, by extension of that, you would expect them to be more similar to cell type specific DHSs. But we haven't actually done the alignment. So that's a good question.
Okay, thank you so much. So there are a couple more questions on the chat box. So Walter, if you're going to be around, you can answer those remaining questions.
For sure. Thank you.
Thank you. So, okay, so the last talk in this session is the towards more realistic simulated data sets from benchmarking deep learning models in regulatory genomics by Eva.
Yeah, okay. Let me just share my screen. Is everyone able to see it? Okay, cool. Hi, I'm Eva Prakash. I'm an undergrad at Stanford working at the Kondaji Lab, and I'll be presenting our paper towards more realistic simulated data sets for benchmarking deep learning models in regulatory genomics. So a common goal in the field is to build classification models that learn to distinguish a positive set of DNA regions associated with a specific function from some background negative set of DNA regions. The resulting models often achieve state-of-the-art performance and are then interpreted for insight into what they learned. We should clarify what we mean by interpretability in the context of this presentation. We use interpretability to refer to explaining the prediction for an input sequence in terms of the contributions from individual bases in the sequence. High positive contribution values indicate that a base drives the prediction upward, and high negative contribution values indicate that a base drives the prediction downward. The inputs to our models are DNA sequences represented as ones and zeros. The input is fed through a series of layers to get the output. The final layer of the model consists of a sigmoid neuron that predicts 1 if a region is accessible in a given cell type and 0 otherwise. So what are the existing approaches to evaluating interpretation methods? One way is to create a completely simulated data set. First, we generate a random background sequence. Then we sample motifs from a position frequency matrix and implant them at random in the randomly generated sequence. In this approach, we have the benefit of being in control of the exact ground truth because we are deciding the motifs and inserting them into known positions in the sequence. However, the problem with this is that these sequences may not be representative of real biological data. Real protein binding is highly cooperative, with many factors often co-binding and affecting each other. And we also don't capture any positional preferences in motif occurrence. Alternatively, we can use a real data set based on real genomic sequences. Here, we can validate interpretations based on prior knowledge of what to expect, and can also validate using orthogonal data sources like transcription factor footprinting data. The obvious pro of this is that it's real representative data. However, it's difficult to ensure you aren't missing any motifs when you don't definitively know the ground truth. So our approach is a hybrid of the real and simulated sequence methods. Given a set of positive and negative sequences identified through functional genomic assays, we run the Homer motif discovery algorithm on the positive set, using the negative set as a background. This will identify the enriched motifs in the positive sequences. We then run the FIMO motif calling algorithm to find instances of these motifs in both the positive and negative sets. It's important to keep track of motif instances in the negative set, as negative sets in real genomic data do contain instances of motifs that may be enriched in the positive set. They are often distinguished from motif instances in the positive set by positional or motif co-binding patterns. Based on the motif calling statistics, we filter out any motifs that do not pass a particular enrichment threshold.
Ava, just a heads up, you're off by 1 click. There we go.
Okay. So for each positive sequence, we then dinucleotide shuffle the sequence to randomize it and implant the motif instances identified in the previous step at their known original locations in the sequence. Note that the original motif match is implanted. We don't sample from the motif position weight matrix. To eliminate GC imbalance between the positive and negative set, for each negative sequence, we randomly choose a sequence from the positive set and dinucleotide shuffle it in order to form the background for the negative sequence. Then, as is done for the positive set, we insert motif matches identified by the motif calling algorithm in the negative set at their original locations in the negative sequence. We do this so that interpretation is not confounded by a GC signal. Finally, we run FIMO again on the positive and negative sequences in order to identify both implanted motif instances as well as instances that may have spontaneously appeared in the dinucleotide shuffled background sequences. The advantages of this approach are that any motifs that weren't discovered by motif discovery are erased by dinucleotide shuffling, so we have some notion of ground truth features. Additionally, having real motifs at their original positions models complex cooperative TF binding patterns. However, a key disadvantage is that we are limited by the motif discovery method, since we could have missed motifs or identified false positives. In this work, we generated our positive set based on 400 base pair regions accessible in 1 of 5 cell types. As our negative set, we used an equally sized set of regions that are not accessible in the cell type of interest, but are accessible in other cell types. Our models were initialized using publicly available weights from DeepSea, Beluga, and Basset, and fine-tuned on simulated data. All models achieved AU-ROCs between 0.75 and 0.85, and AU-PRCs between 0.4 and 0.55 during testing. To calculate AU-PRC for motif recovery, we focus our attention on 10K top-ranked positive sequences, which are likely to contain motifs. We define positive positions as motif region positions and negative positions as non-motif region positions. Following Ku et al., we compute the AU-PRC in each sequence for distinguishing motif regions from non-motif regions using the absolute value of importance scores. We then report the average AU-PRC scores across all the sequences. Now we will give an overview of some of the different methods we compared. Our first approach is in silico mutagenesis, or ISM. In ISM, a perturbation is made to a particular position in an input, and the change in output is computed for all 3 possible perturbations at that position. This is repeated for every position in the input sequence. An obvious drawback to this approach is computational efficiency, though more efficient implementations of ISM exist, such as the fast ISM package. However, ISM also has a saturation problem. So consider the output y, which is a function of the inputs I1 and I2, and eventually saturates to 1. When I1 equals 1 and I2 equals 1, if we perturb I1 from 1 to 0, we won't observe a change in the output, because at this point y is locally insensitive to the inputs. This gives the misleading impression that neither I1 nor I2 is relevant to the output y. To avoid the saturation effect, we'd have to perturb combinations of inputs, but that in turn increases computational cost. Another family of approaches are backpropagation based. You start with a particular output and assign importance scores to the neuron outputs in the layer directly below based on the particular function connecting those layers. You then repeat the process and back down through the layers until you reach the input sequence and obtain an importance score for each base. This is more efficient than ISM because in just one backward pass, you get importance scores for all the positions in an input. There are a number of backpropagation-based approaches, and the most famous is simply computing the gradients of the output with respect to each base in the input. However, gradients don't solve the saturation problem. Note that when I1 plus I2 is greater than 1, the gradient of y with respect to I1 and I2 is 0. So I1 and I2 would once again mistakenly not be flagged as important. A backpropagation-based approach that gets around this is deep lift. Deep lift relies on the concept of the difference from a reference activation, where the reference activation is the activation that a neuron has when presented with a reference or neutral input. For this example, let's assume that a reasonable reference value for I1 and I2 is 0. Now, when we look at the point where I1 plus I2 is 2, for example, the difference from reference is 1, even though the gradient locally might be 0. This is the information deep lift uses to attribute importance to the inputs even when there is saturation. Another reference-based backpropagation approach is integrated gradients. You linearly interpolate from your reference input, which in this case is I1 equals 0 and I2 equals 0, to an actual input, which in this case is I1 equals 1 and I2 equals 1. At each point along the interpolation, you calculate the gradient with respect to the inputs. At the end, you average the gradients across the interpolated points and multiply by the difference of each input from the reference input. You may be wondering what reference we should use in genomics. We recommend using multiple references generated by dinucleotide shuffling the sequence, computing scores for each reference, and then averaging scores across the references. Another backpropagation approach is GradCam. It works like this. First, the gradient of the output with respect to the activations of the highest conv layer are computed. Next, These gradients are averaged along the position axis for each channel to get weights for each channel. Here, alpha k is the weight for channel k. To get the importance at a position I in the conv layer, we then multiply each channel weight by the channel activation at position I, sum this up across channels, and pass the result through a ReLU. The importance at the conv layer is then projected onto the input sequence by linear interpolation. Zhang et al. Used a variant of GradCam, where the gradients are computed at the first conv layer rather than the last. Additionally, conv layer importance is mapped onto the input sequence by averaging over all of conv layer positions whose receptive field overlaps the input position. Finally, the sequence level importance is multiplied element-wise with the input gradients for finer base pair level resolution. Because it's a non-standard version of GradCam, we refer to this approach as the Zhang et al. Method. Now let's discuss some of our key results. Our first finding is that deep lift and ISM tend to perform well. Here are the AUPRC plots. Surprisingly, we sometimes find that ISM perform below deep lift. Looking at an example sequence importance score visualization with the known motif regions boxed in blue, ISM is again seen to miss a motif that deep lift finds. So we suggest two explanations for this. One is that base resolution ISM may be susceptible to saturation effects. For example, mutating a single base may not be enough to substantially disrupt some motif instances. Future work could explore the effectiveness of calculating ISM by perturbing sliding windows rather than one base at a time. Another explanation is that this is a limitation of using a motif discovery method like Homer to define ground truth in our simulations. So perhaps some motifs identified in the positive set are not in fact bound due to missing contextual features, and ISM correctly reflects this. Future work could explore a more advanced simulation that explicitly defines contextual features. Our second key finding is that the GradCam-based method of Zhang et al. Performed poorly in this simulation. So looking at the plots, we can see that the AUPRCs for the Zhang et al. Method are strikingly lower performing than other methods for both models. And looking at an example sequence importance score visualization, we again see the Zhang et al. Method seems to produce more false positives. So remember, the Zhang et al. Method is the product of the input gradients and GradCam at the first conv layer. For comparison here, the input gradients and the GradCam score is shown separately. And it's clear that the false positives are originating from the GradCam piece. And the natural question is why GradCam performs so poorly here. So this is our old GradCam slide. And GradCam computes weights for each channel by averaging the gradient along the position axis. Once computed, the same weights are used for all positions. That means that a neuron belonging to a particular channel will get the same importance irrespective of the position at which the neuron activates or which other neurons are activating around it. In genomics, where motifs can have different effects depending on whether they occur at the center versus the flanks, and also co-binding with other motifs, this assumption may not be valid. This is especially a problem at earlier convolutional layers. In fact, the GradCam paper itself says that localization becomes progressively worse as we move to earlier convolutional layers. And this is seen in the figure from the paper. For context, Zhang et al. Used GradCam at the earliest conv layer, which could explain why it does poorly. Now, you might wonder why Zhang et al. Didn't spot this issue in their own benchmarks. The answer really highlights the importance of a realistic simulation. So this is a description of the simulation method Zhang et al. Used for benchmarking. And you'll note three things. All sequences had a GATA in the center, meaning GATA is not relevant for the classification. Positives had a tau motif, and negatives had no tau motif. So this means the model only needs to learn the tau motif to separate positives and negatives. It can learn this motif with just the first conv layer, and it does not need to pick up any positional or contextual information. As a result, their simulations reveal no problems with GradCam. In fact, to our knowledge, there is no evidence in even the computer vision literature that GradCam outperforms deep lift or gradient times activation when the latter are computed at the same layer as GradCam. So if anything, there is evidence to the contrary, which we discuss further in our paper. Our third key finding is that integrated gradients perform somewhat poorly, at times worse than grad times input. And we can see the poor performance in the plots and also in an example sequence importance score visualization, which is noisier for integrated gradients as opposed to grad times input or deep lift. Our hypothesis for the poor performance of integrated gradients is that when we interpolate between two one-hot encoded inputs, we get fractional values resulting from the interpolation that the network has never seen during training. So we just suspect this, in turn, causes the network to give chaotic interpretations. To summarize, simulations guided by real data are a promising approach for benchmarking interpretation methods. And in our findings, we saw that ISM and deep lift tend to give strong performance. Due to the ignoring of positional information, GradCam-based methods tend to perform poorly, and the performance of integrated gradients can be negatively affected by fractional inputs. So yeah, that's it. Thanks.
Okay, thank you so much for the nice talk. And then I saw Avanti Shirikuma have been answering questions raised during the talk. So let's see if there are more questions. I have a quick question. So you've been looking at this feature attribution methods. Have you looked at this in the interaction, feature interaction, and then what does it mean if you know certain bases, pairs of bases, or triplets have interaction in terms of predicting what you're predicting?
That's a good question. We haven't designed a benchmarking. We've done some simulated data sets where there were feature interactions, and in that case, I think it definitely really helps to have a regression model, because then you can sort of say that when both features are present, the output increases by this certain amount, and then you can exactly quantify the effect of the interaction if you have a simulated regression model. But as for like a simulated interactions that kind of match real data, I think that's hard, because you have to first establish like some notion of like ground truth interactions. And yeah, I suppose if maybe we train a model and maybe through perturbation experiments, we decide that there are ground truth interactions, but then even figuring out what the ground truth interactions is requires a model interpretation method. So maybe if we use perturbation-based stuff, that's one way to identify an interaction, but then it's like, do you trust your model? I think at the end of the day, when it comes to these higher order contextual dependencies, you probably need to design a simulation by hand that maybe matches the statistics of real data as best as you can, but yeah, you need to have much more knowledge about the ground truth simulation if you're going to declare an interaction as a false positive or not.
Right. Okay, thank you. Is there more questions from the audience?
I think Oriol's question was, interesting in case people want to hear the answer to that. So the question was, why are we doing this dinucleotide shuffling followed by a motif insertion instead of just looking at regions where FIMO said that there were no motifs and declaring that as the negative set? And the idea is maybe Homer and FIMO missed some motif instances that are in fact relevant for the output. And if that's the case, then it's possible the neural network would pick up on those motif instances. And then if the interpretation method was saying there's a motif here, then if we were just using FIMO or Homer to define our ground truth motifs, we would tell the interpretation method, no, you identified a false positive, when in fact it might have identified something real that FIMO and Homer just missed. So that's why once FIMO and Homer identify motifs, we then dinucleotide shuffle the sequence to try to destroy anything that they didn't identify, so we can somewhat confidently declare that there are some regions that should not contain motifs because they have been shuffled. That's the idea. How far are we from near perfect prediction of epigenomic signals? Well, I think,
Avanti, can you repeat the question?
Oh, sorry. How far are we from a near perfect prediction of epigenomic signals? Do you think ML will eventually replace experiments? So based on what I've seen, when it comes to the profile prediction models, and Giga from the Kundaji lab has like a really nice paper on this. It's in Nature Genetics, was released earlier this year. The predictions for profiles, that is, the things like the shape of footprints or the distribution of, the shapes of the footprints, those were shockingly good. In fact, sometimes they would, they would do better than replicates, because they would do better than replicate noise at, in terms of their predictions. And they would, you can see that they're denoising the data, and those looked really good. But when it comes to predicting the aggregate signal, those things tend to involve three-dimensional contacts. So I think we're somewhat far away from predicting the strength of the signal. But within a region, if you're told the strength of the signal, predicting the distribution of reads, that's the transcription factor footprints, there we seem to be able to do pretty well in certain situations. So I wouldn't go so far as to say no experiments.
Okay, great. All right. Thank you so much, Eva and Avanti, for the talk and then all these answers. Okay, so it's 11:40, and we are going to have a 20-minute break, and then we will reconvene at 12 at noon with the keynote speech by Regina Barzilay. Okay. Thank you, everyone.
Hi, Regina.
Hey.
Hey, how's it going? Do you just want to check that your screen sharing is working okay?
Yeah, I just want to make sure. I now click the things that I agreed to record it. I never agreed to record it.
Ah, okay. No, that's fine. We can definitely stop. We can stop recording.
It would be nicer to be asked before, not right now. So I don't
Yes.
want it to be recorded.
Yeah, that is fine. I think I do have the power to do that.
So
no problem. And so I'm not doing video just because I'm on not very good visiting my parents in Europe for Thanksgiving and on questionable Wi-Fi.
Yeah. Let me just find it. Okay, this is my. Oh. Okay. Can you still see my slides?
Yes.
Let me just do something else, which is play. Can you see it?
Yes.
When I'm moving the
Good.
slides, can you see the moving slides?
Yes, I see the moving slides. I'm still getting the control bar at the top, but that's not a big deal.
Okay. Okay, I put it on the bottom if it helps better.
So we'll give people a couple of minutes to slowly filter back in.